Hello everyone, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we are going to be talking about X-linked traits because they're a little bit different than the punnett squares that we've been talking about with mono and dihybrid crosses in previous videos. I'll put those videos in the upper right-hand corner in case you haven't seen them because those are important to see before you watch this video. I'm going to take you through the process. We're going to do a punnett square for an X-linked trait, and we're also going to talk about chromosomal mutations in this video as well. Thanks for being here. So the first thing that we're going to notice is in the lower right-hand corner, we've got chromosome pair number 23. Those are the sex chromosomes. 2X is female, XY equals male. The Y chromosome is a little bit unusual. It's very small, and it usually just carries genes for making males male. X chromosomes have a lot more going on there, and they carry some other important genes as well. Interestingly, in females, when you look at somatic cells, usually because there's 2X chromosomes, one of the X chromosomes condenses into what is known as a bar body and basically becomes, well, mostly useless, and the other X chromosome is the one that's expressed. Now that bar body, well, it can change depending on the cell, and so just because you have one X that's a bar body in one cell, well, it can be flipped. The other X can be the one that's expressed in a different cell, and the bar body can be the opposite X chromosome. So females, 2Xs, males, XY. Let's go ahead and take a look at how this kind of plays out when we're looking at how we deal with especially X-linked traits or X-linked characteristics on the X chromosome. All right, so this is a really good example of an X-linked problem that you might see. And we're going to take this to the whiteboard in a couple of minutes here. But you see here, you perform the following cross with the understanding that the characteristic you are investigating is X-linked. And again, we're going to be setting this up because it's a little bit of a different setup. Female is heterozygous. Now females, because they have 2Xs, they can be homozygous dominant, heterozygous, or homozygous recessive, just like we saw with the monohybrid crosses when we looked at punnett squares in a previous video. Males are different. Remember, males only have one X chromosome. The other one is a Y, and they don't share genes between the two, which means we're introducing a new term, and that new term is hemizygous. So males can either be hemizygous dominant, carry a dominant allele for a particular characteristic, or hemizygous recessive, carry that one recessive allele. So we're going to take this to the board. We want to know the couple already knows they're having a boy. What is the probability that the boy will have the recessive trait? Let's go take a look. Okay, so let's take a look at an X-linked trait punnett square. It's a little bit different than what we've done before. So going back to our question, let me go ahead and read that off the screen. You perform the following cross with the understanding the characteristic you're investigating is X-linked. Female is heterozygous, male is hemizygous recessive. So what we have to understand is we have female, XX, male, XY, and that's what we're crossing. So the question tells us the female is heterozygous. Again, just as always, we're going to use letters that are easy to distinguish. So here's our heterozygous female. Male is hemizygous recessive. Remember, there is no trait for the Y. Okay, so there's no allele, I should say, for the Y. Hemizygous means that we're only dealing with the X chromosome, and it is the allele is recessive, and so that's what we have there. We're not putting any letters associated with the Y. This is X-linked only. All right, now, setting up the punnett square is always the same. Here's the female, here's the male. Always starts that way. There's no other way to start this. Here is our female genotype, here's the male. So now what we have is we always are going to have 2X's here at the top, right? And then we're always going to have XY over here. So two females, probability of having a female is 50%. Probability of having a male is 50% if there is random mating. And now we start filling in the R's. So here we have heterozygous female. Here we have homozygous recessive female. Here we have hemizygous dominant male. And here we have hemizygous recessive male. Now the question was, after we do this, the couple already knows they're having a boy, so we don't have to worry. There's basically two ways that a question like this can be asked. The way that I asked it, the couple already knows they're having a boy, so the female outcome here doesn't even matter. All right, so we can just ignore that. What is the probability of that boy having the recessive trait? Well, it's 50%. right? Because we already know we're having a boy. We don't need to worry about up here. If they already know they're having a boy, the probability is 50%. The second way a question like this can be answered or asked is the couple wants to know what is the probability of having a boy or a girl with the recessive trait. In that particular case, the couple doesn't know if they're having a boy or a girl. And so now if I ask the same question, said, hey, you know what? Let's leave the part out where they know the sex of the child. What is the probability of having a boy with the recessive allele? Well, then it's 25% because we don't know if there's a boy or a girl. But if the couple knows if it's a boy or a girl, then you're only dealing with that sex. Hopefully that makes sense. Okay, so that is how you set up an X-linked Punnett square. It's always XX, XY. You're only putting letters associated with the X chromosome. So hopefully that makes sense. Again, the big difference here is that females always have the two X's, males have an X and a Y. Y only have one copy of that particular gene. Now I want to take it a step further, and this is like the grand finale of the Punnett squares in my opinion. We're going to look at an X-linked trait that's almost similar to a pedigree, something we've talked about in a previous video. So in this particular question, let's take a look here. We have a hypothetical question that, well, let's face it, the characteristics don't make any sense. But again, the point here is that we understand how to do these. And so here we have on Krypton, yes, that's right, X-ray vision is an X-linked recessive trait. Okay, homozygous recessive or hemizygous recessive have X-ray vision. Super duper boy is born to a mother with normal vision, but her father had X-ray vision. Super duper boy's father has normal vision. So we're giving you some information about the genotypes and the phenotypes of the family. What we need to know based on that information is, what is the likelihood that super duper boy will have X-ray vision? What if the parents have super duper girl? What's the probability that she would have X-ray vision? So we're going to go to the board. We got to work this through. There's going to be a little bit to do here, but I promise it'll make sense in the end. All right, everyone, this next question, I get it. This is the grand finale of the X-linked traits and really all Punnett squares, generally speaking, because we're dealing with an X-linked trait, but we also are kind of building in this whole idea of a pedigree. Let's go ahead and take a look at the question. So it says on Krypton, X-ray vision is an X-linked recessive trait. So we're going to go here and say that little r is X-ray vision, recessive. Big R, capital R, is going to be normal vision. It says only those who are either homozygous recessive or hemizygous recessive have X-ray vision. Okay, super duper boy is born to a mother with normal vision. Okay, so mom has normal vision. We don't necessarily know her genotype, but we know there's at least one R because normal vision. We just don't know what the second X is, but we're going to figure it out because the next part says, but the mom's father, so grandfather, kind of working our way backwards here, and that'd be XY, had X-ray vision. So, little r. I'm going to show you why that matters in a second. Super duper boy's father, so now we're dealing with the dad. Again, XY. Well, guess what? Super duper boy's father has normal vision, and so that's going to give him a big R. Okay. We need to figure this out because what we need to know is the likelihood that super duper boy will have X-ray vision. So here's the mother's and father's genotype. But we still don't know this. But the only way that we get this big R, so we got to go back to the grandparents. So we're going to do grandfather, grandmother for mom, who we know is at least this. So grandfather's XY, grandmother's two X's. The only way we get this big R, because we know grandfather had a little r, is grandmother had to have at least one big R. So what that means is the grandfather had the only option when grandfather contributed this X to the mom, the only thing he could contribute was the little r. So mom has to be heterozygous. It's the only way that this happens. If grandfather had X-ray vision, the only allele that the grandfather could pass down to the mother is the recessive allele. So now that we know that, we can now fill in the blanks. So here's mom's right here. Dad's got the homozygous dominant allele. So now we can fill in our Punnett squares. So that's girl, that's boy. Right now, it says, what is the likelihood that super duper boy will have X-ray vision? Well, let's figure this out. So we're going to get one possibility as mom provides the X with the dominant allele. The other possibility is mom provides the recessive allele. So for the boy, it's 50%. There's a 50-50 chance that the boy will in fact have X-ray vision. Okay, what about super duper girl? What would be the probability there? Well, for this one, you have two capital R's, so that's normal. And we have a heterozygous potential for a girl. And so there is a 0% chance in this particular case that if they had a girl, that the girl would have X-ray vision. So that's kind of how we figured it out. But we had to go back a couple generations. In other words, we had to kind of incorporate a pedigree to figure out what super duper boy and super duper girl's probability of having X-ray vision were. So hopefully that all made sense. And of course, if it didn't, that's the benefit of having a video. Go back and watch it a couple times. Make sure you understand X-linked traits and this sort of X-linked pedigree that we just did. But it's important to understand with all the Punnett squares that we did, we made a couple of assumptions. One, we had complete dominance. As long as there was one allele present, usually the dominant allele, it was going to be expressed over the recessive allele. And we know that that's not always the case. Sometimes you have codominance and incomplete dominance. And the other thing that we pretty much assumed was that all of these characteristics are independent of one another. In other words, they're not linked. And again, what can happen in real life and nature is sometimes these characteristics can be linked to one another because they're found so close in the genetic code to one another. If you're going to inherit one, you're going to inherit the other. So just be aware that sometimes certain genes are linked together because of their proximity in the genetic code. Now, I did want to take a couple minutes and talk about, well, sometimes things can go wrong in the process of meiosis. Sometimes things can go wrong in the process of mitosis as well, and you can get large-scale chromosomal disruptions. And so I wanted to show you this to start out with, and that is sometimes we have a process called non-disjunction. In meiosis 1 and 2, especially in anaphase, when the homologous pairs or the sister chromatids are supposed to be pulling apart, sometimes that doesn't always happen. And so on the far left-hand side, you see when it happens properly. You have a cell. You've got the homologous pairs up there that separate into sister chromatids, and then eventually you have 4 cells at the bottom with a haploid number. But look what happens in the center and on the right. Here we have a process known as non-disjunction. Up at the very top center, the homologous pairs fail to separate properly, and so what happens in meiosis 1, you have both homologous pairs on the left and nothing on the right. And so what you wind up with is basically all of the cells at the bottom are useless. They're garbage. They either have too many chromosomes, like you see on the 2 on the left, or they have none. So meiosis 1, if you have a non-disjunction there, you're going to wind up with all resulting cells being not the right number of chromosomes. Now on the right-hand side, you can see that meiosis 1 went okay. The homologous pairs separated properly, but in meiosis 2, the sister chromatids in one case did not. And so here, it's still not an ideal outcome, but you can see there's a 50% chance the 2 on the right-hand side have the proper haploid number. The 2 on the left-hand side, one has 2 chromosomes, that's not going to be right, and one has none. In this particular case, that's also wrong. And so at least you have a 50% chance of having the correct number of chromosomes if there is a non-disjunction in meiosis 2. And that's really what I wanted to show you, that meiosis 1, if there's a non-disjunction there, all cells are going to have an improper number of chromosomes, but meiosis 2, if it happens there, at least 2 of the 4 will have the right number of chromosomes. So what does that look like in real life? And so sometimes you get a non-disjunction, and this is an example, Down syndrome, trisomy 21, where the 21st chromosome down there, you see at the bottom, there should be 2 there. There's 3. And what that tells us is there was a non-disjunction that happened in meiosis 2 that led to that. And so Down syndrome is a condition where you have 1 too many chromosomes in that 21st set. Now, this can also happen in the X and Y chromosomes as well, where you get a non-disjunction. And so Klinefelter syndrome is an example of that, where normally you would have either XX or XY. In Klinefelter, you have, again, a non-disjunction that results in two X chromosomes plus a Y. And you can see that there is a whole set of things that happen, phenotypic changes that happen as a result. And here's what can happen if you have a non-disjunction when there are two X's and you only wind up with one instead. Again, that results in Turner syndrome, and you can see that there are a number of phenotypic changes that happen as a result of only having one X chromosome. Now, there are other types of genetic mutations that can happen not just in the X and Y, but in the autosomes as well. And that is something like inversion, where a part of the chromosome is actually flipped upside down. And when these things happen, the regulatory agents that control gene expression may be wrong. The enzymes responsible for reading genes, they can be very, very confused. And so some of the genes on these chromosomes may not be able to be expressed. If the inversion is extreme, the cell may not be viable. This is another example of a chromosomal mutation. This is called translocation, otherwise known as a jumping gene. And that's when a part of a chromosome is removed from one chromosome and is placed inside of another chromosome. We generally have little pieces of RNA that prevent that from happening. It happens a lot more in bacteria when they have chromosomes and plasmids that can sometimes swap DNA. But in human beings, when this happens, again, if that chromosome piece is removed and placed into another chromosome in the middle of functioning genes, that can be a big problem, and that can lead to, again, cells that are non-viable. Another example of a chromosome mutation is a deletion, where a piece of a chromosome has been removed. This is almost always going to lead to a cell that is going to be non-viable because they've either lost a coding region of a gene, or they've lost a control center for a gene, which would be just almost as bad. Gene duplication is another, or chromosome duplication is another potential type of chromosomal mutation. Now, you would think on the surface, like, well, you know what? I had a certain number of genes and I had a certain amount of DNA and that part got doubled. So how could it really be bad? Well, it can be, because sometimes genes are going to be overexpressed, which means we have cells that are in a state of metabolic disarray, and that can also lead to a non-viable cell. Something else I want to consider before we end our introductory conversation in genetics, and that is there are genes and DNA located in other areas besides the nucleus of human cells. Mitochondrial DNA is another type of DNA. We've learned about that before, and we were talking about cells and organelles, and we said the mitochondria at the time had their own DNA, they had their own ribosomes, and their DNA generally codes for genes that are really, really important for mitochondrial health. Mitochondria make ATP, but they do something else that's really important. They actually control, in some cases, whether or not a cell will remain alive, or go through a process called apoptosis, programmed cell death. So if those genes become mutated, sometimes cells are not able to control their own growth and may grow out of control. And scientists are starting to look at whether or not the genes contained within the mitochondria may, when mutated, actually lead to certain types of cancer. Something else that's really interesting about mitochondrial DNA is it's actually inherited from the mother in pretty much all cases. When the sperm fertilizes an egg, the mitochondria are contained within the egg, not the sperm, and so the mitochondrial DNA is actually passed down from mother to offspring. All right, everyone, so that is a short conversation on X-linked traits and genetic mutation in the form of chromosomal mutations. As always, thank you so much for being here. We'll see you next week.